Welcome everybody again. Nice, and we are greatly looking forward to these inspiring speakers.
Hello everybody, my name is Peter Brodin. I'm a pediatrician, pediatric immunologist at the Karolinska University Hospital and Karolinska Institute in Stockholm, Sweden. I appreciate this invitation to give a talk at the ESID meeting, my first so far. And I'm going to talk to you a little bit today about some of the work that me and my team has been doing on systems immunology applied to immunodeficiency and inflammatory diseases. I have a few disclosures of companies that I either collaborate with or advise, and one Cytodelics that I have co-founded together with colleagues in my team. So, systems immunology, in the way that I and my team members think about it, is shifting the focus in immunological investigation from individual cells, individual proteins, to this idea of cell-cell interactions and dependencies, and how such interactions underlie health and disease, how they can be perturbed or dysfunctional in immunodeficiency or inflammatory conditions, and also how we should think about treating not only individual cell types that might be malfunctioning, but actually skew the entire communication network within this system. And so systems immunology means taking cell-cell interdependencies into account. Such immunological investigation has been enabled by a number of technological advances in the recent years, such as single-cell methods to profile genes and proteins in millions of cells, such as high-dimensional plasma protein analysis, allowing us to look at all of the proteins by which cells communicate, as well as the entire genome scale investigations of DNA sequence variants, epigenetic traits, and transcripts. Another really powerful approach that I would like to highlight specifically relates to human immunological investigation, pertains to the analysis longitudinally of patients by collecting samples serially and then investigating changes over time in cells and proteins and genes and transcripts to be able to understand how these things interact and give rise to cellular activation, disease presentation, and so on. And so longitudinal measurements allow us to infer these relationships even when cohorts are very small and cannot fully account for the vast genetic or environmental sources of variation. So I want to talk to you today a little bit about an example in which me and my team members have teamed up with collaborators in the immunodeficiency unit at the Karolinska University Hospital, which are colleagues seeing adult patients with various forms of immune dysregulation and immunodeficiency. And I want to talk to you about a collaborative project we're currently performing on patients with chronic mucocutaneous candidiasis due to STAT1 gain-of-function mutations, and how we have investigated these patients, or begun to investigate these patients and their immune systems using such systems-level analyses. And as many of you know, STAT1 gain-of-function mutations are typically associated with autoimmune presentations, as well as mucocutaneous candidiasis, and also invasive fungal infections, certain viral infections, such as herpes virus infections. And it's believed, thanks to the work of a number of groups, that there is an augmented Th1 and diminished Th17 cell responses. And this is why the presentation somehow phenocopies some presentations seen in patients with autoantibodies to, for example, IL-17, such as APS-1 patients. And so what I'll tell you about today is just an example or a little vignette of how investigations using systems level methods in such patients can teach us new things about this disease. So what we've done in these patients together with our colleagues in the hospital has been to collect blood samples longitudinally, as I mentioned earlier, pre and post treatment. And the treatment in this case have been using a JAK inhibitor. And what we've measured at each time point is a global assessment of cell composition using mass cytometry, CyTOF. We've stimulated whole blood directly at blood collection with prototypical antigens such as LPS, BCG, and IAV, flu. And then we've analyzed transcriptional responses by bulk mRNA sequencing. We've also analyzed plasma protein analysis and collected bulk transcriptomic data without stimulation. And so just to give you small examples of what we might find from these investigations in such patients, I'm showing you 3 sample visits, and these are immune cells analyzed by mass cytometry. And what you'll see here is these are 2-dimensional single cell plots using a projection analysis of single cells projected onto a 2-dimensional map based on phenotype. And these are only CD4 T cells above and CD8 T cells here. And at visit 1, the patient has received no treatment. And at visit 2 and 3, the patient is currently on a JAK inhibitor treatment. And what I want you to take away from this figure is that there is a dramatic redistribution of CD4 and CD8 T cell populations after treatment with a JAK inhibitor, suggesting that T cells are completely remodulated when given this treatment. We see from the stacked bar charts on the right showing proportions of immune cells that there is a shift in especially the CD4 compartment from naive to more central and effector memory T cells after treatment, as well as a expansion of memory Tregs after treatment. And I should say that the clinical effect of this treatment has been very good in this particular patient. So a lot of these symptoms have recovered, and this is an immune correlate of that recovery. And so pretty dramatic redistribution of immune cell phenotypes in CD4 and CD8 T cells. But maybe a little bit less known from previous work is that there are many other immune cell compartments that are also affected by the JAK inhibitor in the STAT1 gain-of-function patient. And for example, the monocyte compartment shows a pretty strong or a large proportion of cells within this activated pro-inflammatory monocyte, and that is sort of silenced after treatment. And in the NK cell compartment, there is another complete shift in phenotype away from CD56 low towards a more CD56 bright phenotype, more regulatory NK cell phenotype in STAT1 gain-of-function after JAK inhibitor, really suggesting that the treatment effect is broad, involves many different cell types, and can inform us, of course, about the mechanism of action of this treatment in these patients, and also explain a little bit why the symptoms improve beyond just the T cell redistribution. Another thing I want to highlight is a more general aspect of the work we're doing, where we are benefiting from this beautiful consortium called Milieu Intérieur, headed by Louise Quintana Mercy and initially Mathieu Albert and now more Dara Duffy in Paris, at the Pasteur Institute. And here is a elegant paper from this consortium looking at the variation in transcriptional responses to prototypical whole blood stimulations, showing and in a large cohort of healthy individuals in France. So what this shows is that when 1,000 individuals are blood samples have their blood samples stimulated with one of these prototypical antigens, there is a transcriptional response that is very similar across that vary mostly between type of stimulation given, but there is also a gradient, an inter-individual variation here that can be obviously caused by environmental or genetic variation. And we believe that this is a beautiful reference cohort onto which we can overlay patients with suspected or confirmed immunodeficiencies, and then we can investigate more how these deficient individuals vary in contrast to this healthy individual. So this is exactly what we have done. And so what I'm showing you here is in bright colors, pink, blue, and yellow, are the control subjects, which are the same individuals as from the taken from the milieu interior cohort, and it's the same data. And then overlaid on top of that healthy control data is PID patients, different kinds of patients with different mutations and different defects, overlaid on top of these patients. And as you can see, there are some of them, such as these purple ones down here, which have a nearly normal response as compared to the control patients stimulated with the same BCG agent here. But there are also other patients with a suspected or confirmed immunodeficiency that respond in a completely different manner from their corresponding control population, allowing us to really look into the specific transcripts that can explain this variation and help us understand disease mechanisms better. So this is ongoing work. And then back to the same type of stimulation done in this patient with the STAT1 gain of function mutation. This is 1 out of 3 patients we've looked into. And what I'm showing you here are effect sizes, meaning changes between flu-stimulated and unstimulated. And then these are ranked transcriptional modules such that initially there is a dominance after flu stimulation in the STAT1 gain-of-function patient without treatment to upregulate transcripts involved in immunoglobulin responses and plasma cell responses, antigen presentation, and monocyte-related transcripts. But these are all decreasing after treatment, and instead we see a more normal upregulation of type 1 interferon responses after JAK inhibition in patients with STAT1 gain of function. And as you all understand, when stimulating with flu, a type 1 interferon response is a prototypic response that you expect to see. So we believe that these types of analyses contrasting patients to healthy individuals with the same prototypic stimulation, we believe is a way to better understand disease mechanisms in primary immunodeficiencies. So now I will switch gears and I will talk about COVID-19 and some of the inflammatory syndromes that have arisen as a consequence of this infectious disease, and some of the work that me and my team have been doing in this context. In general, what we are trying to do is to use systems level analyses to compare patients with different presentations, all induced by this one virus, SARS-CoV-2, which is mild to severe COVID-19, MIS-C, and now more recently, long COVID. And I'll start with talking about two different papers that have come out of our group and our collaborators' work in the last few months, looking at either acute COVID or MIS-C and Kawasaki disease. So if we start with patients with severe acute COVID-19, when we profile their immune cells in longitudinal manner by mass cytometry, and I'm showing you one example patient here, what we saw was that over time after admission to the hospital, there was a change in multiple cell types, and I'm highlighting eosinophils here because these were a little bit unexpected and pretty dramatic changes. What we see is that 2 to 4 days after admission in this patient, there is an expansion of an eosinophil population expressing CD62 ligand, probably induced by interferon gamma, as based on prior literature, and probably homing to the lung, based again on what's known about eosinophils expressing CD62 ligand. And what's intriguing here is the time point, because we know that right around the first week of after admission is when patients are most likely to develop ARDS, acute respiratory distress syndrome. And this expansion of eosinophils with this phenotype precedes that perfectly. Another event that happens right around the same time point is an expansion of monocytes expressing the CX3CR1 receptor, the fractalkine receptor. And these cells are intriguing because we know that the fractalkine system is involved in cells trafficking along the blood vessels and then extravasating into inflamed tissues. And so this opens up a therapeutic potential opportunity here. So we know that hyperinflammation in the lung is a critical component of COVID-19 and the severe cases, and probably a cause of acute respiratory distress syndrome and even more mortality in these patients, one of several important causes. And there are a multitude of treatment options attempting to mitigate this inflammatory state, such as IL-6 inhibition, IL-1 inhibition, and so on, interferon gamma inhibition. But all of these treatments are aiming to mitigate an already existing hyperinflammatory state in the lung. What if instead we could try to prevent cells from extravasating from the blood into the lung and thereby mitigate or prevent even the hyperinflammatory state? So one option based on our data to do this would be to block the fractalkine receptor. And together with the Swedish company Cancera, which have a drug in called CAND567, which is a inhibitor of the fractalkine receptor, we have initiated a clinical trial in COVID-19 patients to try to prevent severe COVID-19 and hyperinflammation in the lung by preventing immune cells from leaving the bloodstream and going into the inflamed lung using the fractalkine system. And these patients are adults with signs of lung involvement, but not already in need of a ventilator. And we aim to randomize patients into either the fractalkine receptor inhibitor or placebo, and then treat them for 7 days and then do follow-up studies. And the primary endpoint is to minimize the need for oxygen support and lung involvement, as well as obviously monitoring the immune system over time. Another thing that became clear when the COVID-19 pandemic rolled along over Europe and the US was that children who are typically very mildly affected by this virus can present with a very severe hyperinflammatory condition called MIS-C, multisystem inflammatory syndrome in children with COVID-19, or PIMS-TS, which is another name of the same condition. And we had already at the time an ongoing study together with Paulo Palma's lab in Rome looking at Kawasaki disease children. And these were children sampled in 2017 and 18, so prior to the SARS-CoV-2 virus. And so we decided to try to profile this new condition and the immune system, both with respect to antibodies, immune cells, and plasma cytokine levels. And the first thing we asked was whether the hyperinflammation seen in children with MIS-C would be the same as the hyperinflammation seen in adults with acute COVID-19. And it turns out when we looked at 112 unique plasma proteins that is not the case. These are discretely different conditions where the MIS-C patients are here, the adults with acute COVID is here, and Kawasaki disease patients with a few overlaying MIS-C patients are here. There are a number of cytokines that differ, and this has been recently published in Cell, so I refer anyone to the publication for more details. In fact, what we find is that MIS-C patients are sort of at the intersection between Kawasaki disease patients and their inflammation and children with mild SARS-CoV-2 infection, which are overlaid by healthy individuals, suggesting that fitting with the idea or the finding that SARS-CoV-2 is usually very mild in presentation in children, not even distinguishing them from healthy children. What the main differences are that Kawasaki disease is associated with a very strong activation of IL-17, IL-17A, and a very high abundance of IL-6 in circulation, while the MIS-C patients have lower amounts of those cytokines. We also assessed serological responses in these patients, and we found that MIS-C patients, just like children with SARS-CoV-2, produced anti-SARS-CoV-2 IgG antibodies in equal or similar amounts. But there were differences in their abundance of antibodies to common cold coronaviruses. And in particular, we found that MIS-C patients clearly were devoid almost of such antibodies to common cold coronaviruses, while both Kawasaki disease patients, children with mild COVID-2 infection, as well as healthy children had such common cold coronavirus antibodies. And it's important to note that the MIS-C children in this cohort were typically older than any of the other groups, which might explain this difference, but this requires further investigation. Finally, in the end, we decided to look for autoantibodies in MIS-C because of the fact that the pathology suggests an autoantibody mediated pathology with which responds very well to IVIG as well as other immunomodulatory therapies, and resolves quite quickly in these patients, even the very severe patients. And we report in this publication a number of possible autoantibody targets that will require further downstream analysis for validation to investigate immune complex formation as well as tissue activation at the target site. But some of these candidates were particularly intriguing when we looked at over 9,000 different autoantibody targets using these whole proteome arrays. One particular group of protein that I would like to highlight are these casein kinases, because casein kinases are widely distributed in these different tissues. And in a recent study from Nevin Krogan's lab, spearheaded by Boztug et al. In Cell, this group reported that casein kinases were particularly upregulated, induced by SARS-CoV-2 infection in human cells. And in fact, they were able to show that in the cell membrane of infected cells, there are tentacle-like structures that carry viral particles and presumably infect nearby cells. And these are mediated by casein kinases. And in our analysis, we find that antibodies to casein kinases are strongly induced in patients with MIS-C, more so than in any of the other disease conditions we've looked at, potentially suggesting that these autoantibodies are important in the pathophysiology of this condition. So with this, I would thank a phenomenal group of researchers that I work with at the Karolinska Institute in Stockholm, as well as colleagues in the immunodeficiency unit at the Karolinska University Hospital, and colleagues in Rome and in Stockholm who collaborated on the MIS-C study. With this, thank you very much, and I'm happy to take any questions.
Good morning. I'm Ronald Germain from the Laboratory of Immune System Biology in the National Institute of Allergy and Infectious Disease at the National Institutes of Health in the United States. And today we'll be talking about imaging immunity. I have no disclosures, and I want to start with, I hope, what is not a provocative statement, but one that I think is accurate, which is that basically all immune function occurs in a complex lymphoid or parenchymal tissue environment. And given that, we cannot explore it looking only at isolated or extracted cells or molecules. And so to look in the tissue context, we need to image, and we would like to have both dynamic information, but also high content information. And there are a whole series of questions that we'd like to answer using these types of imaging approaches. For example, what are the cellular dynamics that are involved in normal, but also pathologic immune responses in vivo, and how do those temporal or spatial or dynamic components relate to function? How is tissue microanatomy, the organization especially of lymphoid tissue, involved in promoting host defense or autoimmunity or immunodeficiency or tumor destruction? What are the molecular signals that guide the positioning of these cells and their interactions and their effector functions? And what happens as the immune system is interacting with epithelial or parenchymal or tumor cells in diverse body sites? And so the first method I want to talk about today is multiphoton dynamic imaging. It can give you this kind of temporal or dynamic information we'd like to collect, but for technical reasons, it's very restricted, usually to fewer than 5 parameters at any one time. It's difficult to do signaling measurements. Calcium works well. There is an NFAT reporter, but otherwise, not many good methods exist for doing this. The range of observations is seconds to some hours before you lose tracking of cells, and it's not directly applicable to human studies, except in certain explant situations. The basic technology, especially in the mouse, where it's used most often, is to surgically expose the tissue of interest. And that's true for everything other than skin, which means you need to worry about whether the person doing the experiment is introducing artifactual inflammation in what is presumed to be a normal tissue environment. And there are important controls that need to be run for that. Assuming that those controls are properly run and you collect the information by scanning the laser in 3 dimensions, and then continuing that over time to get stacks of imaging data, you wind up playing back these movies at about 200 to 300 times real speed. Here, looking at naive T cells moving in the paracortical region of a lymph node. And you have this impression that these colored objects, these cells, are free to move in this empty space. But you should always keep in mind that this is really the environment those few labeled cells are moving in. And you should never by a conclusion that's reached by an imager based on what he and she does not see. You should always worry about what's in the black. And so we and others over the last 2 decades have used this for many purposes in the mouse, looking at lymphoid movement in the lymph node, observing that T cells move on these fibroblastic reticular roadways to promote interaction with dendritic cells, to look at the dynamics in germinal centers, study the capture of bacteria by protrusions of dendritic cells through the gut epithelium, looking at dynamics in the kidney, looking at the myeloid components of granulomas in liver, as I'm showing you here, and also the lymphoid components. With a bit more difficulty, looking at dynamics in infected lungs, looking at myeloid cell innate responses to damage in the ear skin, looking at T cell responses in the same environment during delayed type hypersensitivity responses, looking at the movement of cells such as osteoclasts in the bone marrow, and more recently, as we've reported, looking at the very acute response of resident macrophages to tissue damage to prevent excessive neutrophil collateral tissue damage. And the problem is, as I said, you're limited to only a few parameters, hard to execute this in humans, but even in the experimental mouse, we're looking at this very small volume of a mouse lymph node, and we miss a lot. And so to overcome that, we'd like to do histochemistry, and we started doing this to see what was outside of our imaging field, only to realize that we were in an era in which 2 or 3 color immunofluorescent staining was the standard, but everybody in the lab was walking down the hall to do 12 to 15 color flow cytometry because you needed multiple markers to phenotype different cell populations. And that's just emphasized here in looking at one of these current kinds of panels for looking at typing blood cells, for example, many, many more parameters than you would see in a typical standard protocol immunofluorescence or immunohistochemical analysis. And so I want to spend the rest of the time today talking about what I call multiplex static methods. They don't capture the dynamics of the cells like 2-photon imaging does, but the parameter space now can almost routinely exceed 40 to 50 parameters. I'll show you even more than that a bit later. You can probe with the right antibody signaling and post-translational modifications. You can look at nuclear localization of transcription factors. You can study both proteins and RNA using RNA-fish approaches. And you can use this easily with ethically collected human material. And so our own version of this particular approach we call histocytometry was developed in the lab by Michael Gerner. And we were able to stain in up to 14 colors. That's a bit on the extreme side, but typically 7 or 8 colors at a time. Improve the image with deconvolution, compensate as you would do in flow cytometry for channel spillover with the different fluorochromes, and then use software tools that would enable us to create segmented cellular objects with all the associated fluorescence for the markers that they actually had. But of course, that is actually the same as flow cytometry data, cellular objects with associated fluorescence. And so we are then able to go in and gate in the way you would do in flow cytometry to look at complex phenotypes, get quantitative information about those cells, but because we also have done this in the tissue context, see where each of those cell populations resides in the tissue context. And I will show you later, but this method works very well with human samples. I'll give you an example where I show how it works with antiphosphoprotein and anticytokine antibodies. So this allows you to look at phenotype, signaling state, and function along with spatial positioning in the tissue. And so just as a quick illustration of how this works, if we just look in the middle here, we can gate around these B220 stained cells and call them B cells and make them blue. Gate around the CD3 positive cells, call them T cells and make them red. And FlowJo gives you a nice sagittal section of a mouse lymph node with peripheral B cell follicles and central T cell zone. We've done very quantitative analyses to see whether this is an accurate method in that regard compared to flow cytometry. And if we look at naive cells, we get virtually identical results, whether we extrapolate the numbers from a section by histocytometry or we take apart the lymph node and do classical flow cytometry. But if we begin to look at other cell types, and in particular dendritic cells, this small wedge that's very hard for you to see of the number of dendritic cells we get by flow cytometry after standard extraction procedures. But this is how many we quantify using the histocytometry. It's about a 20% overestimation because of difficulty of segmenting very irregularly shaped cells like dendritic cells, but it's not this many-fold difference. And the reason is that those dendritic cells are very tightly adherent to the fibroblastic reticular cells that I showed you in an earlier movie, and most digestion protocols do not fully remove them, and you throw them away when you do your nylon screen treatment to get single-cell suspensions. And so I need to point out, and I'll repeat this again later, that this underrepresentation of certain cell types, particularly myeloid cells and a number of stromal components, applies to any method of extraction. And CyTOF, single-cell RNA-seq, and all of these methods, as powerful as they are not the ground truth for what's actually happening in a tissue. You get much more accurate reporting and fewer false negatives, that is, the absence of things that are actually there when you do this by imaging. And so we've used this in one of our earliest applications to look at all the different dendritic cell subsets that had been defined at the time in a mouse skin draining lymph node. And the interesting feature that went further than early reports from Bernard Mallison is that different dendritic cells live in different regions of the lymph node. And in particular, classical, what are now called CDC2s that primarily present via class 2 to CD4 T cells, were highly resident in the periphery of the lymph node, whereas CDC1s that are known for cross-presentation via class 1 to CD8 T cells were much more in the central T cell zone. And so that helped us begin to add to earlier data we had developed about how immune tissue or lymphoid tissue is organized to promote what is essentially an incredibly inefficient system, which is adaptive immunity, where you need to have very rare antigen-bearing dendritic cells interacting with even rarer antigen-specific CD8 or CD4 T cells, or those CD4 cells interacting with antigen-specific B cells. If you think about this, where you need a ternary mix, a dendritic cell and a CD4 and a CD8 T cell, the likelihood that they would come together by random motion in the volume of a lymph node is virtually zero. And so the question is, how does this really work, and how has evolution adapted the system to get efficiency out of this inherently tricky system? And so recently, Antonio Baptista in the lab, using these imaging methods, came up with an additional piece of information that had been missed over the years in doing classical histology. What he showed was to repeat the work that Mike Gerner had done, that I just recounted to you, that the CDC2s live in the peripheral region that is far away from the center, as plotted here, of the lymph node, and the CDC1s are more centrally located. And when we looked at naive CD4 and CD8 T cells that had up to this time been considered to be randomly mixed and randomly moving in the paracortex of the lymph node, it turned out that was not correct. There is a clustering or a density increase in the CD4 T cells in the peripheral region near the CDC2s, which is, of course, a match between the class 2 presentation preference of the CDC2s and the class 2 recognition specificity of the CD4 T cells. And conversely, the CD8s, which were less biased in their distribution, but clearly abundant in the center of the lymph node, were matching in the location of the CDC1s. I'm not going to go through all the data since this is public, but what we were able to show is that the oxysterol receptor EBI2 is what controls the peripheral positioning of the CD4 naive T cells. And if that is eliminated in a knockout animal, now there's no bias in the distribution of the CD4 and the CD8 cells. And that has a very dramatic impact on cell-mediated immunity. We're able to look at early events in T-cell, dendritic cell interaction by what we call a clustering analysis, looking for multiple T cells to be localized on a dendritic cell platform. We don't see much of that 3 hours after immunization. But by 9 hours after immunization, we see these clear clusters here in red, which are the wild type cells that we're tracking. But in blue, you see an absence of that clustering among EBI2 deficient cells. And this is consistent with the fact that they're further away from the dendritic cells, and it takes them longer to move throughout the lymph node environment to find the relevant dendritic cell. And this delay turns out to have a very dramatic effect on host immunity. One of the other fellows in the lab, Anita Gola, had developed, along with Adrian Hill as a graduate student, a very efficient way to protect against liver stage malaria. There's a systemic adeno-antigen priming and then a boost with IV nanoparticles that creates tissue-resident memory CD8 T cells in the liver that protect against sporozoite infection of hepatocytes. And the critical thing to remember is that in this model, the CD4 T cells are required during priming to provide help for the CD8 cells, but at the time of challenge, depletion of CD4 T cells has no effect. All the effector functions mediated by the prime tissue-resident memory CD8 cells. And when we look at an EBI2 deficient animal, we see this dramatic loss in protection against sporozoite challenge. Now, that animal is lacking EBI2 in all cells, and there could be many things wrong in that environment. So we repeated that experiment where we transfer EBI2 deficient CD4 T cells with wild-type CD8 cells into RAG deficient animals. So the only EBI2 deficient cells are the CD4 cells, and we see an even greater loss in protection under those conditions. And other experiments show that EBI deficient CD4 cells, once you do get them primed, are precisely as good as wild-type cells. So this deficiency is not an effector deficiency. It's a delay and a decrease in the CD4 response that's discoordinated now from what you need to help the CD8 response, and you lose essentially all protective immunity. And so to summarize this part, our imaging data show that at the steady state, there are very asymmetric or non-random distributions of different dendritic cell subsets and also of naive T cells in a lymph node. There's a match between the class 2 presenting CDC2s and the class 2 recognizing CD4 T cells. So they bring these cells that need to interact into close proximity to improve the efficiency of response at the early times after antigen drainage into the lymph node. It's mediated by EBI2. And if you disrupt this, you lose immune response potential at the cell-mediated level upon vaccination, or as I'm not showing you here, there also were problems in clearing helminths under these conditions. And so this goes to a larger schema that we built up over the years using these types of imaging methods that talk about the fractal biology of lymph node structure and function. The antigen-specific lymphocytes and the dendritic cells are not just localized everywhere in tissues, but they congregate in the lymph node. So you concentrate two reactants, and we know that that improves reaction rates and completion of reactions. Within the lymph node, we now know there are different zones, and the dendritic cells and the T cells themselves are co-localized just in the T cell zone. So we've increased this volumetric packing. We then understand that there's this fibroblastic reticular cell network that exists in the lymph node, and our data showed that the T cells move on this network, and that's important because it turns out the dendritic cells are also attached to this network. So we've created an additionally smaller subvolume that helps promote the interaction of the T cells with their relevant partner cells, the dendritic cells. There's still a random walk on those FRC fibers until at least some antigen is introduced in the system and at least some T cell finds a relevant dendritic cell. But once that happens, we now get the production of chemokines that now bias the walk of other T cells in that environment. And here, the CD8 T cell will find this interesting and engaged an antigen-bearing dendritic cell about 8 times more efficiently than this irrelevant dendritic cell that's otherwise the same distance away. And again, if you interfere with this, you lose virtually all CD4 help for CD8 T cells. And then, as I just shown you, there is an asymmetry in the distribution of dendritic cells. And that's matched by an asymmetry in the distribution of the T cells to further bring the relevant interacting partners together to make what is otherwise an inefficient system operate effectively. Now, that method, histocytometry, even up to 12 or 14 parameters, grossly underrepresents the parameter space that we now understand for immune cell subsets, as well as many different parenchymal cell types, structural elements, and in the case of tumors, the transformed cells. And so we've now developed a new method that lets us more extensively probe that complex environment. And Andrea Radke led this and was helped in this by Evelyn Kandoff, who's now an MD PhD student at Vanderbilt. And what they developed is called IBEX. There are other iterative methods. I'm not going to have the time to talk about the other ones, but in this particular method, we've worked out ways to get very good tissue adherence to slides. That's not a trivial part if you're going to do iterative imaging. We then have also worked out ways to increase how rapidly we can get full penetration of even thin sections with antibodies to decrease the time this whole cycling takes. We then image the stained section in 4, 5 to 7 or 8 parameters. We then bleach the fluorochromes, and we've validated about 20 different fluorochromes. We know their precise behavior in this bleaching solution, no matter what antibody you connect them to. So it's a very reproducible event. And then we go back and restain and reimage and iterate this many times. We validated somewhere close to 200 different antibodies to immune and non-immune targets in human tissues that range from liver, lymph node, thymus, lung, gut, kidney, not shown here, so that we're able to apply this across many, many, many different tissues. And so here is a human lymph node stained in 82 different parameters, which to us, as far as we understand it, is sort of a record. If you're interested in records, only 4 of the colors are shown here. If I turn them all on, you actually would wind up seeing basically white, because blending all the colors basically gives you white light. But now you have a problem. If you were to do this, if you had done CyTOF or flow cytometry, and you had even 40 parameters, the iterative binary gating to identify specific cells based on their clusters of relevant markers is really quite a complex exercise. Now think about doing that at 50 or 60 or 70 parameters, where you also care about the spatial organization of the cells. And so in order to deal with this without having to do this all manually, Nishant Thakur in the lab has come up with a machine learning algorithm that's based on compressing all the channels into 3 channels and grading them by intensity, so we can deal with dim or medium or bright cells. And this is a reversible algorithm, so we now can take that complex staining pattern I showed you, and we wind up with this pseudo-colored image where I have to stress the pseudo-colors are not individual fluorescent stains, but a cluster of markers revealing an individual cell type. And so if we look in the germinal center that I'm showing you here, this algorithm says that these 4 parameters define naive B cells. That's what we recognize them at, and they're all outside the germinal center. These 6 markers define germinal center B cells, and they all show up inside the germinal center. These are the dividing B cells that are enriched in the dark zone, and these are the TFHs that are enriched in the light zone, and so forth. So we're taking this 80-parameter data set and now able to go parse out these very complex phenotypes and subsets in an automated way, and I can't show you for time today. We're able then to do a spatial analysis to see who's living near whom and what's disturbed if you're not looking in the normal tissue. Now, that's still not enough, because any given section subsamples imaging space, so we've gone on to develop a method that allows us to do this multiplex staining in clear tissue to get 3D images. And so here's a mouse lymph node in 5 colors, where you see the beautiful branching arborization of the HEVs when you see it in this 3D view. But as we go to the section view, you wouldn't understand that 3D anatomy if you only saw these individual cross sections. But we also can look and phenotype all the other millions of cells in this large volume that would be the equivalent to a core biopsy that you might take in a patient. And so I want to end by telling you about some unpublished data having to do with combining the histocytometry and the 3D multiplex to understand Tregs in tissues. And this is work of a terrific fellow in the lab, Harry Wong. We had published previously that IL-2 producing conventional CD4 cells occur even in the presence of Tregs. That is, Tregs do not prevent the activation of autoreactive cells, left open the question of how autoimmunity is averted by Tregs. What Harry did is to turn the difficult task of looking for IL-2 into one of identifying the autoreactive cells based on PD-1 expression. And this just shows you an example of a PD-1 positive CD4 cell that's not FOXP3 positive. And the important point is the same number and distribution of these PD-1 positive cells is seen in germ-free mice, so we know they're autoreactive, and in concert with our earlier data, they're pSTAT5 negative. We then can develop spatial statistic methods that let us look at the how this PD-1 positive autoreactive cell is localized in space relative to other cell types, in particular Tregs. And this interesting result is that compared to non-PD-1 positive naive CD4 cells, these PD-1 cells that are the autoreactive cells live in small microdomains or islands in which there's an enrichment of regulatory T cells. Because we've done multiplex staining, we can do the equivalent of the kind of analysis you see from CyTOF or single-cell RNA-seq, and we get different clusters that correspond to different states of the regulatory T cells. And when we look, what we see is that those cells that are activated with high numbers of markers, I'll show you in a second, are the ones that are tightly associated with the autoreactive cells. And these Tregs are dividing. They're Ki67 positive, 25 high, CTLA4 high, FOXP3 high, and pSTAT5 positive, identifying them as highly suppressive effector Tregs. And development of this effector phenotype and their presence in the microdomains requires IL-2 from the conventional cells. So these Tregs are not operating in a prospective manner. They're operating in a negative feedback mode to constrain immunity. And those microdomains are generated by local proliferation of the Tregs. So there's also feed-forward amplification that helps ensure that you are constraining these autoreactive cells. It still didn't answer precisely how you achieve a control. So we turn to a system where we control the exact timing of when the autoreactive cells see self-antigen by transferring some transgenic cells at numbers that achieve physiological levels in the immune system, 10 or 20 cells in an entire lymph node. When we do that, we can analyze them quantitatively using our cleared 3D imaging. So here, all the red cell objects are Tregs. These very small numbers of purple and green cells are the autoreactive cells and control cells. And when we look, we get a surprising result. Over the first 3 days after those cells see their self-antigen, they actually proliferate. So Tregs also don't prevent these cells from proliferating under these conditions. However, by a week later, they've basically disappeared. And we put this together in a model in which we have putatively lower affinity conventional cells and higher affinity Tregs. They both see their self-antigen. There's a small amount of 25 put up on the conventional cells. They make IL-2, but the greater 25 on the Tregs steals the IL-2. That imposes this proliferation and microdomain formation of effector Tregs that help constrain the conventional cells. IL-2 does not actually drive proliferation. Cells are driven into proliferation by MYC upregulation through TCR signals alone, but when you steal IL-2 and they don't sense it during proliferation, they die, leaving you the clearance. We've gone on to mathematically model this whole situation with the Tregs and confirm the modeling by experimentation and shown that very small changes, so 50% loss of CTLA4 expression, for example, the equivalent of CTLA4 hets in humans, or a 40% reduction in local density of the autoreactive environment in terms of the Treg density, imbalances this fine-tuning and leads to escape of the conventional cells from this control to promote autoimmunity. And this obviously has important implications for understanding how genetic polymorphisms, like EQTLs with only twofold changes in gene expression, can contribute to autoimmunity and for attempts to modulate Treg function. In the end, we're trying to combine the methods that I've talked to you about today for imaging immunity, using intravital imaging to look at dynamics, then fixing the cells in place, moving on to multiplex 3D imaging, and then correlating the cells we analyzed deeply for phenotype back to the original dynamic imaging. And by playing the movie backward, we can relate the dynamic behavior of cells to their subsequent phenotype. And this is all part of a larger effort to use both the dynamic imaging, the lower plex cystocytometry, the high plex IBEX, our computational approaches for data analysis, our 3D imaging, and in work I haven't shown you today, in combination with in situ RNA, really to understand exactly how the immune system is operating in the tissue context. Thank you very much.
Hi everybody, my name is Michael Menager. I'm a group leader at Imagine Institute in Paris, in the Necker Hospital campus. And so today I would like to show you how we've been using single-cell omics coupled with machine learning to try to revisit patient stratification in autoinflammatory diseases. First, I would like to start by showing you and presenting you my team, introducing my team. We are studying inflammatory response and transcriptomic networks in disease. One side of the team is working on understanding the interaction between HIV and dendritic cells, try to understand why there's not an optimal type 1 interferon response mounted by disease to the virus. And the other side of the team is working on analyzing rare genetic disease at the single cell levels in interferonopathies, which are diseases with an excess of type 1 interferon. And then a third arm of the team is composed by computational biologists, where we're trying to use machine learning to predict transcriptomic pathways involved in inflammatory response, both in response to HIV sensing and in case where there's rare genetic mutations leading to autoinflammations. We have two positions open for computational biologists, so if you're interested, please contact me by email. That being said, I have nothing to disclose. And so I will start today's presentation by introducing the concept of how we're using single-cell omics to stratify patients. So with some of my colleagues at Imagine Institute, we're working on primary immunodeficiencies. Here, we've listed 6 different primary immunodeficiencies. And genes have been identified, so monogenic disorders in those diseases. What's interesting is several genes can be mutated, and they seem to be in the same functional pathways for each disease. So one clinical presentation, you can have several genes mutated once at a time. And then with the progress of next generation sequencing and so on, we've also discovered that the picture could be more complex because you could have different mutation in the same genes. Here we could take the example of FOXP3, and different mutation could lead to different primary immunodeficiencies with different clinical immune signs, such as autoimmune cytopenia or lupus, depending on the mutation. So the picture is becoming more complex because one gene can lead to several clinical presentations. This is where we thought that we might benefit to couple this genetic diagnosis with maybe some kind of diagnosis at the molecular level. We also think it's important because when you're testing a treatment based on a cohort of patients sharing clinical signs, oftentimes some patients are not able to respond to the treatment. And this is where also this absence of response could also be due to some molecular heterogeneity at the level of patients, which is also another point to try to stratify patients at the molecular level alongside clinical and genetic diagnosis. This is a particular case of interferonopathy. So disease characterized by an excess of type 1 interferon, inflammation, without any absence of infection, which is damaging to the immune system and the body. Each gene represent a gene identified as a monogenic disorders for different interferonopathies. And here you can see very heterogeneous clinical spectrum of clinical manifestation depending on the genes mutated, with at least in interferonopathies 2 things in common: inflammation of the skins and some problems in the central nervous system. So the solution for that, the proposed solution, is to try to stratify the patient at the molecular level. And for that, we've been using, since 2017, the technology from 10x Genomics based on microfluidics, as you can see here. And basically, you have a solution, aqueous solution with cells mixed with oil, and that's creating a water droplet. And in each droplet, you will get a cell, and then a magnetic beads with poly-DT tails. And after lysis, this bead is going to be able to capture the messenger RNA expressed in the cells. So we've been coupling this transcriptomic analysis with barcoded antibodies, which are tagged not by a fluorochrome, but they are tagged by a piece of DNA, a barcode, and a poly-A tail, which means they can also be captured in the droplet. And after library preparation, we can see the transcript expressed in the cells, but also the antibodies that were at the surface of the cells. We've been working in most of our pathologies with cells extracted from the blood, peripheral blood mononuclear cells, PBMCs, and we're working with 20 frozen PBMCs for each samples in order to be able to analyze up to 8 samples at the same time by single cell libraries. So per sample, we can generate 10,000 single cell libraries up to 8 different samples at the same time. Now, with single cells, we are not able yet to uncover all the genes mutated. Sorry, not all the genes expressed. We're only able to detect the top 15-20% of the genes the most expressed in each cells. So the way people have been using single cells is based on the top gene expressed. Cells are clustered in a 2-dimensional graph, such as those UMAP plots. And what's been shown in the blood is most of the clusters found in the blood correspond to the classical immune cell types: B cells, T cells, NK cells, monocytes, and so on. So now the idea would be to compare those clusters coming from the blood of healthy blood donors with cells coming from the blood of patients, here patients with a loss of function mutation in SAMHD1, which drive an interferonopathy. If we can identify a cluster of cells that would be enriched in the patient or found only in the patient, we would call those pathological cell clusters. And then we could extract from that transcriptomic signatures and try to look for new biomarkers. We could also test if here we're talking about the Aicardi-Gouttières syndromes, AGS. You have different genes mutated, SAMHD1, TREX1, MDF5, among others, leading to the same syndromes. Then we could check that, although at the clinical levels, we're talking about the same syndromes, at the molecular level and the single cell levels, are we talking about the same pathological clusters? And then we could also compare patients with different genes mutated and different syndromes, here SAVI versus Aicardi-Gouttières syndromes, and this time maybe looked for homology at the molecular level, whereas there could be a difference at the clinical, in terms of clinical signs. Finally, we've also been investigating if we could follow the benefit of a treatment on the evolution of such pathological cell clusters to use single cells to use single cells to see the therapeutic response. So in addition of gathering information for each of our patients at the clinical level, the genotype, generating single cell transcriptomic, proteomic, and epigenomic level, we have also projects where we would like also to combine with lipidomics, metabolomic, and microbiome in order to also see the impact on the environment on those diseases. So now what's happening is with all those different omics, you're generating a tremendous amount of data, big data. And so the idea is how to integrate all those multi-omics together in order to try to have, at the end, one multi-omics network. And for that, we've been starting to use machine learning approaches to try to infer those multi-omics networks. So in terms of multi-omics data integration, where do we stand in my team? So we've been collaborating with Rich Bono and Dan Littman in New York. And so for our HIV project, we've been able to use RNA sequencing and epigenomic, but at the bulk level, not at the single cell level, to be able to create a network where we connected together more than 22,000 genes and predict the change of activity of more than 500 transcription factors in order to try to find new genes and new pathways involved in interferon response and dendritic cells maturation after following HIV infection. So I will encourage you to look at this paper if you want to know more about the technique we've used for network inference. And so now we'd like to reproduce the same type of approach at the single cell level regarding machine learning. I would like to mention this project that is led by my colleague Frédéric Rioloca at Imagine Institute, which was recently financed by the French National Agency of Research, ANR, Attraction for Autoimmunity Inflammation through RNA-seq analysis at single-cell level for theoretical innovation. So in this project, we're starting with 6 different primary immunodeficiencies, a total of 250 pediatric patients. We have also we will have the equivalent of 250 pediatric controls. The idea is to perform the different single-cell multi-omics that I've talked about, and then re-stratify a different patient at the molecular level based on pathological clusters, based on molecular signatures. Then in the second phase, we'll use machine learning and network inference to try to identify functional pathways that could be applied to each of those subgroups in order to see if we could identify new molecular targets, meaning get into personalized treatment and medicines. And also, we're also thinking of drug repurposing. So now I would like to show you some examples of the type of data we already obtained using single-cell omics. And before that, just a word of the single-cell hub at Imagine. So with my team of research, we've been performing single-cell experiments since 2017. And then recently, thanks to fundings from Région Île-de-France and Imagine, we've been able to open core facilities, single-cell at Imagine, where in combination with other core facilities, bioinformatics, genomic, and data science, we can perform and analyze our single-cell data. So if you have project and would like to use our core facility, we are open to all academic and industrial, so you could contact me, and we're also looking for computational biologists on this core facility. The first example would be an example of identification of one of those pathological cell clusters. Here, it's a collaboration with the team of Frédéric Rioloca. We are looking at a family where there's an activating mutation in STING, a gene involved in DNA sensing, and this leads to autoinflammatory syndromes called SAVI. And you will see in this family, in addition of a classical sign of a SAVI, such as inflammation of the lung and skin vasculopathy, there's also a very severe sign of autoimmunity with lupus identified. This individual here, the father, has a twin brother, and interestingly, although they're carrying the same mutation, and it's a monogenic rare disorder, the twin brother doesn't have those autoimmune signs, or at least they are not as severe, and so he doesn't have any lupus. The father had a daughter, and she's carrying the same mutation, and this time the daughter also developed some severe autoimmune sign. And so we were fortunate enough to have frozen PBMCs from the daughter before and following 2 treatments, JAK-STAT inhibitors, to decrease autoinflammation. We have also samples can be compared to a sample from the on-call before and after treatment. Unfortunately, we were not able to obtain samples from the father who died. And as controls, we have also PBMCs from the mother and the aunt of P2, which are not carrying the mutation and, of course, not developing the disease. So in total, we have 8 samples, and we have processed, we have generated libraries using 10x genomics for those 8 samples. And for data analysis, we've been using the algorithm SERAT, or the pipeline SERAT, developed by Raoul Satija at the New York Genome Center. So basically, we generate the library with 10x genomics, with sequence on a NovaSeq. We use Cell Ranger to process raw data. And then we are keeping cells with at least 500 genes and less than 20% of mitochondrial genes. We perform them several normalization and scaling steps. And then using canonical correlation analysis, we are able to integrate the clusters coming from our 8 different samples in the same UMAP. And so this is what you see here. We are looking here at more than 40,000 PBMCs coming from 8 different samples, and they are segregated in different clusters using our protein expressed at the surface and transcriptomic markers. We're able to identify B cells, myeloid cells, mate cells, NK cells, combination of naive and activated CD8, CD4 cells. And here, what was interesting is cluster 12. Those seems to be T cells, but they were hard to classify. And so here, when we looked at what's the contribution of each samples to this cluster, here in abscess, this is the percentage of total PBMCs. You can see that cluster 12 is composed mostly of cells coming from the patient P2, so activating mutation instinct combined with Lupix sign. And after treatments, cells doesn't seem to cluster anymore. And our uncle with no lupus sign doesn't seem to have cells enriched in this cluster. So here, maybe we've identified a pathological cluster that could be correlated with the presence of Lupix sign in the P2 patients. So once we have such cluster, we're asking to derive the molecular signatures of this C12 cluster. So this is cluster number 12. Here you can see the top 50 genes, which correspond to the signatures, and with a high expression in yellow mapped in 100 cells for each cluster. So you can see that the signatures seems pretty unique to cluster 12, except that the signatures was also found in cluster 19, which first I thought was a problem because I wanted to extract a signature specific to this pathological cluster. So now let's look at cluster 19. So this is a tiny B cell cluster, and cluster 19 seems to have the same profile as cluster 12, highly enriched in the patient with Lupix sign and decrease after treatment. So we think now we have identified 2 pathological clusters, one derived from T cells, one derived from B cells, and we've extracted their molecular signatures. So if you looked at genes upregulated in the clusters, you can find 132 genes specifically upregulated in these clusters, and you can perform some enrichment and pathway analysis here using EnrichR. And we clearly start seeing T cell activation, pathways with T cell exhaustion, and also central tolerance. So which is give us clues now that we're investigating to try to maybe better understand the appearance of those Lupix sign. We also identified downregulating genes, and here we see a clear NF-kappa B inactivation that seems to be linked to the cells belonging to this cluster. So now I would like to show you a second example, which is, can we follow the impact of a treatment at the single cell level? Now we'll talk about the pathology of Aicardi-Gouttières syndromes, which is also characterized by an excess of type 1 interferon. This project was performed with my colleague and his team, so with my colleague Yannick Rowe at Imagine Institute. And we are still working with cells from the blood, PBMCs. We're comparing this time 2 different unrelated healthy blood donors with the cells of a patient with a loss of function mutation in SAMHD1, leading to an excess of type 1 interferon and these Aicardi-Gouttières syndromes, before and 6 months following a treatment aiming to decrease type 1 interferon. We are using the same technology to realize a library and to analyze single-cell data. We've here plotted 8,000 cells from 4 samples in this UMAP, identified all different clusters. And one thing you can do, you can also now split the cells per samples. You have here cells belonging to the patient before and after treatment, and of 2 healthy controls. The first thing we can observe here is following the treatment, there's the appearance of specific cell cluster, myeloid T and B cell cluster only seen in the patient after treatment. We also identified that seems to be a decrease in mate and activated CD8 cells in our patients, which doesn't seem to be too much corrected by the treatment as compared to our healthy controls. Since we're looking at a pathology characterized by an excess of type 1 interferon, we've also used signatures to look at the score expression of 272 known genes to be upregulated by type 1 interferon. And if we're asking where are the cells responding to type 1 interferon, with no surprise, this is targeting the myeloid monocytic cell cluster. So now if we zoom on this cluster, we also see that there's a strong type 1 interferon response in the patients, but not in the controls. Interestingly, after treatment, there seems to be appearance of a new cluster of cells, which seems to have less type 1 interferon-enriched genes. So now we're focusing on this. So what's interesting, cluster 14, 9, and 2 are composed of cells coming from the 4 samples, patients and healthy controls. And they're clustering together because they belong to the same cell type. Whereas cluster 10 here at the middle is composed of dendritic cells, CD16 and CD14 monocytes. But all cells belong to the patients after treatment. And so which means the cells are clustering not based on their cell types. But here, when we looked at the interferon score signatures, cluster 10 seems to be the cells where, after treatment, the myeloid monocytic cells have decreased type 1 interferon. So here, thanks to single cells, we've identified precisely 277 myeloid monocytic cells in which we can observe a downregulation of type 1 interferon signatures following the treatment. And so the same, now by looking at the genes downregulated and upregulated, we are now looking to try to understand why type 1 interferon has been downregulated in those cells, but not others, and what are the pathways, the transcriptomic pathways linked with this downregulation of type 1 interferon. Now, so far, we've been working single-cell omics in rare monogenic disorders. Can we use single-cell omics for more complex multifactorial inflammatory disease? Here we are switching to a pathology called juvenile idiopathic arthritis, so leading to inflammation of joints in child and with no apparent cause known. And so the same, in collaboration with my colleague Frédéric Huloka, we've been working with frozen PBMCs, so cell extracting from the blood of healthy controls and GIA patients. And here we will look at 4 patients, 1 patient with no known genetic causes, 2 patients where we've identified mutation in the same genes, they are monozygotic twins, and we think the mutation participates to the phenotypes, and another patient with a mutation in the different genes. So we've performed single-cell libraries, we've analyzed them, we're mapping now 40,000 PBMCs coming from our 8 samples, we've identified the clusters. Now we're separating the cells belonging to the healthy controls on the left, and the cells aggregated from the 4 GIA patients on the right. Looking at cluster bias, we identified a loss of activated CD8 T cells in our patients, an increase in monocytes, and the appearance here of tiny clusters which seems to be derived from monocytes. If we focus on this monocytic-derived cluster, which now are the cells highlighted in red. So most of them are monocytes, and we can also find some of them in the T cell compartment. If we looked at the composition of samples in this cluster, once again, we call it a pathological cluster because more than 90% of the cells of this cluster are coming from 4 GIA patients. So we think here we've identified a common pathological cluster to 4 GIA patients. As we did before, we've been extracted the molecular signatures of this cluster 13, and now we're using those genes. We're trying to see if those could become new biomarkers for GIA patients that could be tested by qPCR, or for example, flow cytometry using a surface antibody. And we're also exploring the molecular pathways associated with these genes. Interestingly, and maybe not as a surprise, there's an enrichment for genes known to be involved in rheumatoid arthritis, which is the adult form of GIA. And we also, it's also linked with inflammatory response and defect or switch in metabolism, which could also give us clues what could be linked to this pathology. And interestingly, we've kind of confirmed now the pathological aspect of cluster 13 because this is almost the only cells where there's a very high expression of those two chemokines, CXCL4 and CXCL7, which have been reported in 2016 to be able to define early stage of rheumatoid arthritis in synovial macrophages. So which means we can detect at the transcriptomic level and at the single cell levels potential markers of early sign of the establishment of a disease. And so with the time I have left, I would like just to show you one last very interesting example, which is so far unique. Another collaboration with my colleague Frédéric Rioloca. This time we're looking at loss of function mutation in an immune checkpoint regulator, CTLA4. Without CTLA4, that leads to lymphoproliferative syndromes of autoimmune clones. And so here we've profiled PBMCs of a father and mother from two different marriage. Father and mother number 2 are healthy carriers. The patients with profile is cells, but after treatment. So you can treat those patients with abatacept, which is mimic the effect of CTLA4. And the disease is so severe that it's not easy to get samples before treatment. And in red, you have markers looking at T cells. So globally, after treatment, we don't see major difference. And for patient 1, we obtain samples after treatment. You can see here that looks pretty similar to controls of the other patients, maybe with problems in monocytes. But very interestingly, we also collected at the time without clinical signs as a control is PBMCs before any treatment. And now if you look at the PBMC profile, the profile looks totally abnormal compared to the treated or the other patient after treatment, which suggests that potentially here, thanks to single cells, we could have already some clues at the molecular levels of establishment of a disease before the appearance of the clinical signs. So now we are further investigating this very interesting case and trying to find some more. So with that, I would like to thank you for your attention. Thanks my different source of fundings, people in my lab, people in the lab of Frédéric Rioloca, which help us on those projects, Imagine Core Facilities, and people from Yannick Rose lab and Nadine Serf-Bensoussan's lab. Thank you.